Okay, next up is a startup from UHN, and going back to the theme of TAP membership hospitals interacting with us in the UTEST program. Epiloid is another example. They're a UHN startup that is enabling epileptic drug treatment through the use of their unique machine learning algorithms. So over to Epiloid Technology with Mark.
Thanks, Hassan. Hi, everyone. I'm Mark. I'm the co-founder of Epiloid, and our mission is to revolutionize the drug development process in neurology. And currently, that drug development process is very risky. Over 90% of all neurology drugs fail to pass the clinical trials to make it to market. The leading factor to that failure rate is that mice are sadly not humans. And this means that our standard preclinical studies poorly translate to human patients. Even when a drug is promising enough to enter clinical trials, there's no way of knowing which participants will respond best. And for diverse neurological diseases like epilepsy and depression, which can manifest in many different ways, this leads to unclear clinical trial results, costing pharmaceutical companies billions of dollars. Now, clearly, there's a need for a better solution. And luckily, all is not lost. There is a new technology called human brain organoids. And these are essentially 3D miniature brains that can be grown in a dish from either human stem cells or from as little as a patient's blood sample. And this means we can now more easily get human brain tissue to test in a lab. And that's exactly what we at Epiloid are doing. By using our proprietary methods to grow organoids to replicate human disease, we're able to provide early insight into how a new drug will interact in human brains before clinical trials. We can use the genetic backgrounds of patients to accurately replicate disease and find the right patient populations to target. This means pharma companies have better insight into which drugs to pursue, saving them billions of dollars. How we're doing this is through our unique combination of 3D tissue engineering and machine learning. First, we produce dozens of these organoids using our proprietary techniques to model disease. Next, we track how the electrical activity in the brain tissue changes in response to direct testing with the new drug. And finally, we process those recordings through our AI algorithms, which are able to predict which kinds of future patients are most likely to be positive responders. Currently, the contract research market for neurology is exponentially growing. We're aiming to establish our first beachhead in epilepsy, which has a North American market of over $600 million. And based on our growing list of partners, we project an initial revenue of just over $600,000. The way we're doing this is through offering per compound service contracts, ranging from preclinical to clinical studies. And from that earliest point of a drug's life, we started ensuring that a candidate drug is safe and effective at reaching a desired neurological target. Our main flagship service is in providing efficacy measurements of a drug in human brain tissue before it ever touches a patient. Moving into the clinical trials, we can directly test the clinical trial participants before they interact with the drug. This de-risks toxicity and tailors the dosing recommendations. And finally, our data can be put to use in selecting the best patient segments to target, stratifying clinical trials, and assessing orphan drug candidates early. We've already validated each of the pillars of our technology. We've proven that organoids derived from patients retain their epileptic response. We've profiled how organoids respond to existing anti-epileptic drugs. And we've proven that we can use our algorithm to predict which drugs would be best in treating mice. Now, we're putting our technique through its first major validation study with our pharmaceutical partner, Avicanna. In terms of our intellectual property, we have a provisional patent underway supported by the University Health Network around our method of generating epileptic organoids. We're also currently co-developing a multi-electrode array with the team behind another University of Toronto startup, NerveX. The device we're making together has been tailor-made to our specifications for high-throughput organoid testing. And last but not least, our AI algorithm is currently being protected through trade secret. What's next for us is to develop new intellectual property around organoid development and targeting new disease areas, including depression, Alzheimer's, and mitochondrial disorders. And here's how we stack up to our competition. The obvious gold standard is tried and true animal testing. And while these are usually pretty great models of a disease, they clearly lack the human element to make smart decisions about drug development. There are some contract research organizations that can provide human spinal cord tissue, but these aren't relevant to modeling diseases of the brain. and other companies doing 3D tissue engineering can make brain organoids, but they're focused only on toxicity studies. And that's really where we shine. We are the only ones who have an inducible human epilepsy model in a dish. And this means we can now get functional efficacy testing of new drugs for epilepsy. In addition to our list of early adopters and partners, we're working towards exciting opportunities in developing new intellectual property for recording from organoids with the team behind NervX, and have identified stem cell technologies as an eager partner for licensing opportunities. We've also made connections to several champions of our cause in the drug development sector, including Drs. Amza Ali and Justin Grant, in the community outreach, such as Gary Collins, the president of Epilepsy Canada, and in neurology through Dr. Talia Field, one of Canada's top 40 under 40 neurologists. Our roadmap to success continues with our validation study with Avicenna. We're currently refining our drug processing pipeline to be better able to serve the partners who've reached out to us for our services. In the near future, we aim to complete a patient-derived database of organoids collected from the epilepsy patients here at the University Health Network in Toronto, as well as taking our production of organoids entirely in-house. In about 5 years, we expect to expand our offerings to other neurological diseases. Currently, we've received just over $300,000 thanks to our partnership with Avicenna, but are limited in the number of drugs and organoids we can test at a time. With an investment of $1.5 million, we're looking to invest in the hardware to accommodate new partners. And with a raise of $4 million, we'll be able to take our production of organoids entirely in-house and set ourselves on the path to achieving our targets for future years. This is the team making it happen. My background is in 3D tissue engineering, epilepsy, and signal processing. Rohit Singla is an MD PhD candidate out of the University of British Columbia with a background in machine learning. And Dr. Peter Carlin is an epileptologist and neurologist at UHN. Our team of scientists is also working very closely with key business advisors, Gary Collins, again, the president of Epilepsy Canada, and Michael Vaughan, who's a biotechnology entrepreneur, as well as several of the incubators in the Toronto area. And I hope you agree that Epiloid is positioned to revolutionize the drug development process in neurology. Thank you.
Great pitch, Mark. I remember when you talked to us back in May of this year, you were talking about Avicenna, and I think Curtis and I were very surprised at how, a group like yours was interacting with a group like that, straight out of the research lab. How did that whole partnership form, and where do you see that going?
Yeah, absolutely. There was a great opportunity to work within that space. Avicenna is a company that is very interested in the use of medical cannabinoids. And we've had a great relationship with them in terms of validating the science behind cannabinoid use. So there was a call to be able to work with that. And our existing key opinion leader and Dr. Peter Carlin, who's one of the lead neurologists in Toronto, was able to make that bridge with this up-and-coming pharmaceutical company. And since then, we've had an excellent working relationship. They really want to have that validation in terms of how cannabinoids can be put to use in best serving the epileptic community. And that's really where we want to show that our science works.
Have you tested this outside of epilepsy in a variety of other neurological conditions?
Our personal focus has been on the electrical activity with epilepsy, and that's really where we've been driving that focus. I'm keeping a keen interest in the space, and we're excited to work in the depression and mitochondrial disorders, and that's where our next steps are generating from. So we've gotten some preliminary insight into that, and it looks very promising. However, there's still a lot of research work to be done, and these are still very young models that continue to surprise us and give us new insights. So.
And I think a follow-on question to that is, why did you go for epilepsy first versus other neurological disorders?
Yeah, well, epilepsy is really our bread and butter, and that's where our expertise is. So we're able to really confirm that this is a valid model of epilepsy that we're able to see in these organoids. A lot of the partnerships that we've been able to make with other pharmaceutical companies as well as other key partners have been because of that connection with Dr. Peter Carlin, within UHN and some of our connections in that space. There's a clear pathway forward for us and some of the partners that we've already worked with. We've already gotten traction in that epilepsy space, and we see that that's our key beachhead for making a splash.
Okay. You mentioned you have plans to build a database of patient organoids. How are you going to do that?
Yeah, we've been working within the University Health Network here in Toronto. So we've already collected some patient samples, and some of our validation studies have already worked with patients with rare encephalopathies, where we've seen that they have a specific genetic mutation. So we already have a couple that we've generated. We have a research ethics board approval for being able to collect blood from the variety of different patients that are coming through UHN, which are some of the most severe cases for epilepsy. And with Rohit being part of the Vancouver ecosystem of hospitals there, we're also branching out into that. The long-term plan would be to partner with the biobank and to be able to really have access to a lot of patient information, patient blood there that we'd be able to leverage for growing out these organoids in a larger scale.
I hope I don't butcher the next question, but can you look at mechanism of action related biomarkers? I think that's the question. MOA related biomarkers?
Yeah, it's tricky to really be able to assess memory. There are very specific organoids that can be generated that mimic sort of particular regions of the brain, which each have their own function. At this stage, the models are pretty young, and they best mimic sort of that developing brain, where memory is kind of a more established brain structure. So it's quite difficult to model those kinds of memory structures at this point. That is definitely on the innovating side of organoids. And as that technology continues to develop, that's something that we want to keep an open mind for and being able to address that, because there's a lot of disease areas that are really underserved in the kinds of models that they can provide right now. And that would be something that's on our radar as organoid technology continues to develop.
Yeah. It looks like you're working with a few pharma companies who are paying for your services. How did you acquire them? How are you planning to acquire new customers?
Yeah, absolutely. We've actually had these pharmaceutical partners reach out to us for our services. Again, a lot of that is because of our clout in that epilepsy space, and Peter Carlin is recognized as an innovation leader in this field. So really the problem for us is that we're not able to service all of the customers that have come to us. So I'd say our acquisition cost for customers is zero at this point. And these small to medium pharmas seem to be the ones that we've generated the most traction. They're keen on this edge on their other competitors that we can provide through organoids. And this gives them great insight into an assurity that their drug will work as they take it into the more expensive phase 1 clinical trials.
Okay. Next question. You anticipate $4 to $5 million in recurring revenues by 2024. Can you comment on how you anticipate that revenue? Which customer is the low-hanging fruit?
Yeah, absolutely. There are a few customers that we've identified, including Cerevel, who's out of the States. They're a key player. There's UCB as well, which is a major pharmaceutical player that is in the epilepsy space that is accessible to us. We've already started some of these dialogues with them to be able to see if there's an opportunity to work with them. There are a few opportunities there that are underway. Part of that revenue is also in potential licensing partnerships. As I mentioned, we've already begun speaking with Stem Cell Technologies, and there's an interest there in being able to license some of that technology off with them. They really have a lot of the infrastructure of being able to take that on and leading a certain aspect of that, where our focus can be more tailored to these small to medium pharmaceutical companies.
Mark, as the final question from me, what's your ultimate vision for this whole platform and company?
I can absolutely see this being a technology that can grow into something where not only is it serving the pharmaceutical partners that need this insight, but this can really help a patient to be able to get that insight into which drugs best serve them. Right now, the best gold standard that physicians have is sort of a shotgun approach of try a drug and it doesn't work out. And if we're able to provide a data-driven approach to saying this is the right pharmaceutical compound for a particular patient or patient group, that would provide that peace of mind, that ability to be assured that the pharmaceuticals that you are taking and you're ingesting are really working for you.
Well done, Mark. Thank you for your time. Appreciate it.